Okay, so now we're going to talk some more about membrane transport, but we're going to talk about the second principle. And the second principle is to ask, is energy required? And so let me switch to a demonstration that I do. Normally I do this in class with students, but let me show you what I do here with these. So normally these would be students. This here is to represent a membrane. You'll notice there are some little holes in this membrane. So that would either be spaces in the phospholipids that molecules could get across, or it could be a protein that would allow it across. But usually I have students stand on one side of some sort of physical barrier that has some holes. And then I ask them, well, how comfortable do you feel over here? Do you have enough room? What if you did jumping jacks? Would you still feel like you had enough room? And usually it's quite crowded. And so I ask one to come across. No problem. Okay. And usually I'm able to get it across to where there's close to equal numbers on each side. And in this case, everybody's got a sufficient amount of room, as much room as possible, and they're generally pretty comfortable. Okay. Now I'll ask students to go the other way, and I'll say, okay, well, please go across the membrane. And this one might grumble a little, but it'll go across. Okay, well, how about you? Will you please go across? And okay, goes across. Now by the time this one gets there, that student really doesn't want to go across. And I have to really beg a lot to get this student to go across. And this one, I can barely get this student to go across. It's too crowded. They don't want to go over there. Here, look how much room they have. Okay. So with this one, I've got to put a lot of energy in to get it to go across. Now, energy is the key term here. So to go where there's a high concentration of, in this case it was students, but molecules, to a low concentration doesn't take much energy. That's easy to do. Okay. But to go the opposite way, from a low concentration to a high concentration, you have to put energy in to get it to happen. Okay. Now that's not an analogy in real, or that is an analogy. In real life, molecules don't care if they have space, they don't have feelings, they don't have nervous systems. So what's actually going on? Well, it turns out that molecules have random motion, and this is called Brownian motion, and they're just, and there's a video of it in the homework, I believe, where you can see it, and they're just kind of bouncing around randomly. Now, if they bounce around enough, eventually they're going to hit a hole and go through. All of these molecules are bouncing around. Well, it's more likely, if you have a whole bunch of molecules on one side, it's more likely that at some point this molecule will hit the hole and go through. Over here, I mean, you've only got 2 chances for that molecule to happen to hit it and go through. So this is why going from a high concentration to a low concentration takes no energy. But to go the opposite way, you got to have some energy and grab it and pull it across, because that's not likely to happen on its own. Okay. All right, so let's merge some vocabulary words into this concept. Okay. So we have 2 types of transport. We've got passive transport, and we've got active transport. Okay, with passive transport, you are always going from a high concentration, and concentration just means amount. You're going from a high concentration to a low concentration. This is always, always, always the case, and therefore no energy is required. Now with active transport, it's the opposite. You're going from a low concentration to a high concentration. and energy is always required. And usually that energy is in the form of ATP. Now, so that is always the first question you ask yourself, is you always ask yourself, is energy required? And if it is, you separate it into passive and active. Now there's going to be subtypes of passive. Okay? Passive subtype is, one type is simple diffusion. That means that no protein help is needed. Okay? You can also have facilitated diffusion, and that means that you do need protein help. For active transport, you always need a protein. Okay. So these are the rules. Remember, passive, if it is passive, these things are going on. Or you could do that backwards. If no energy is required, then it must be passive. Okay? And if energy is required, it must be active. Okay? So once you ask yourself about energy, then ask yourself about protein help and get it to the specific type that you're talking about. Okay. And so I believe those are the principles of transport there. So what I'd like for you to do is to use this knowledge and go through and see if you know it well enough to fill out this chart. Now wait for a minute on vesicles and vesicular transport. Okay? We'll come to that in a minute. But see if you can fill that part out. Now, one thing we need to talk about are these channels and carriers. So channels are not used in active transport. Neither are carriers. But in passive transport, both are used. Okay, and you can relook at that video and put a description in your own words about how channels and carriers are different. Pumps, they work similar to carriers, except for they need ATP, so therefore they are used in active transport, but not in passive. Okay, and so again, for this description, they work very similar to a carrier, except they need ATP. Okay. Now, I do want to talk a minute about this energy for transport needed for active transport. Now, there's this molecule called ATP. Again, these are these chemical structures. You don't need to memorize them, but what I do need you to notice is that ATP stands for, and let me go ahead and write this down, stands for adenosine triphosphate. So that's ATP. And you'll see the ATP. Okay. Tri means, this part right here, means 3. So if you look at the structure, we've got 1, 2, 3 phosphates next to each other. Now, these phosphates are all negatively charged. You should notice these negative charges. And what did we say about negative charges and positive charges? Well, negative likes positive, but it turns out negative hates negative. And so what this means is that these 3 phosphates hate being together. They're kind of like people at a family reunion that don't really like each other, but they're forced to be in the same place together for a while. Most of us have maybe had that experience at some point in our life. And so these three things hate each other. So how is this used for energy? And again, this is your ATP, T standing for tri, which is 3. Is it turns out that this phosphate breaks off. So say you're at a family reunion and you've got these three people butting heads. Well, one of them decides to go into another room. Now, this is the key to understanding how ATP provides energy for transport. This phosphate will go and stick onto your protein, your enzyme, your transport protein. When it does, it changes the shape of the protein. And remember, structure always changes function. So putting this phosphate on and off of these transport proteins changes their shape, and that's what allows them to carry molecules across. Okay? So that's how ATP provides energy for transport. Okay? And here you can see it. We've got the sodium-potassium pump. It takes 2 sodium molecules out of the cell while it takes 2 potassium in, and it is burning ATP into ADP. Notice that D stands for di, D-I, and di, D-I means 2. So here it has 2. It's lost this third one. So what I'd like for you to do here is in your notes packets, there are 3 different types of transport. I pulled these from your book, but I'd ask that you try looking at these without looking at your textbook first and see if you can figure them out. And I'm going to pull up some bigger images of this. So what type of transport is it? Okay, so let me walk you through the first one if you're having problems thinking about it, and feel free at any point to pause and see if you can get it on your own. Okay, so what you should first notice is how to interpret this cartoon representation. So these little gray dots are representing carbon dioxide. Okay, and this orange line right here is the boundary of the cell. So if you want to think about what type of transport is it, the first thing you have to ask yourself is where's the high concentration and where's the low concentration? Well, if you just count the dots, there's a high concentration inside the cell and a low concentration outside the cell. So if we look at these gray dots, we're seeing by the arrow shows which way it's moving from high to low. And we said if we look back at our rules, high to low is passive. So right here we can say passive, but we want to get more specific because if you notice on our list, there's 2 subtypes. We can have simple diffusion or facilitated diffusion. Well, how do we know which one it is? Well, you have to look at what the molecule is. It's carbon dioxide. And so, if we go back here to this part, we already learned this is one of our examples. Carbon dioxide is small, it's hydrophobic, and it does not need protein help. We look at our definition. No protein help needed, so simple diffusion, and so we come here and we say it's passive and specifically simple diffusion. Okay. Now, I'm just going to help you interpret the other images, but I want you to see if you can get them right and email your instructor if you need help. So again, orient yourself. Here's the plasma membrane, these little red dots with the tails. These are our transport proteins. And you notice we're taking glucose, bringing it into this protein, and moving it across. And so this is frame 1. It shows you've got 4 glucoses here, and you're moving 1 across. You've got no glucoses here. And on this side, we've moved it across from one side to the other. So I want you to think about again, which type of transport is it? Now let's again look at this one. Now to interpret this figure, these little hollow things show where it started, okay? And then the arrow shows the direction it moved, and now it's showing where it ended, okay? So again, the orange is potassium. It started on this side and moved here. So you say, where was potassium high? Where was it low? Which direction did it move? Did it meet a protein? The same for sodium, okay? This was where it started, these little empty places, and we've now moved it over to this other side. Another clue for the picture could be ATP being converted into ADP. So think about what are the clues there, what do they mean, and which type of